Tonight's lecture is over Chapter 24, the Digestive System, and this begins on page 941. The digestive system contributes to homeostasis by breaking down food into forms that can be absorbed and used by body cells. It also absorbs water, vitamins, and minerals, and it eliminates wastes from the body. The purpose of this chapter is to identify the anatomical components of the digestive system as well as their functions, discuss the neural control of the digestive system, compare and contrast mechanical and chemical digestion, discuss the effects of aging and disease on the digestive system. The digestive system includes the digestive tract and its accessory organs, which process food into molecules that can be absorbed and used by the cells of the body. So there are four main functions of the gastrointestinal tract. The first one is ingestion. This process involves taking foods and liquids into the mouth, commonly called eating. Digestion. Digestion is the process of breaking down ingested food into small molecules that can be used by the body cells. In mechanical digestion, the teeth cut and grind food before it is swallowed, and then the smooth muscles of the stomach and the small intestine churn the food to further assist the process. As a result, food molecules become dissolved and thoroughly mixed with the digestive enzymes. In chemical digestion, the large carbohydrate, lipid, protein, and nucleic acid molecules in food are split into smaller molecules by hydrolysis. Digestive enzymes produced by the salivary glands, the tongue, the stomach, the pancreas, and the small intestine catalyze these catabolic reactions. Absorption is the movement of products of digestion from the lumen of the digestive canal into the blood plasma or the lymph plasma. Once absorbed, these substances circulate to cells throughout the body. A few substances in food can be absorbed without undergoing digestion. These include vitamins, ions, cholesterol, and water. Elimination. Wastes, indigestible substances, bacteria, cells sloughed off from the lining of the digestive canal, and digested materials that were not absorbed in their journey through the digestive canal leave the body through the anus in a process called defecation. The eliminated material is termed feces or stool. So this is figure 24.1 on page 942. This shows the organs of the digestive system, and the accessory organs are indicated in red. Organs of the digestive canal are the esophagus, stomach, small intestine, large intestine, and anal canal. Accessory digestive organs include the mouth, teeth, tongue, salivary glands, pharynx, liver, gallbladder, and pancreas, and are indicated in red. The GI tract is a long tube that is opened at both ends for the transit of food during processing. The named portions of the tube include the esophagus, the stomach, the small intestine, the large intestine, and the rectum. Accessory structures. Accessory structures are not part of the GI tract, but they do contribute to food processing. The accessory structures include the teeth, the tongue, the salivary glands, the liver, the gallbladder, and the pancreas. So this next cartoon is on page 944, and this is figure 24.3. And this shows the layers of the GI tract. Variations in this basic plan may be seen in the esophagus, stomach, small intestine, and large intestine. There are 4 layers of the digestive canal. From deep to superficial, they are the mucosa, the submucosa, the muscular layer, and the serosa. The mucosa, or inner lining of the digestive canal, is a mucous membrane. It is composed of a layer of epithelium in direct contact with the contents of the digestive canal. It also has a layer of connective tissue called the lamina propria, and it also has a thin layer of smooth muscle. The submucosa consists of areolar connective tissue that binds the mucosa to the muscularis. It contains many blood and lymphatic vessels that receive absorbed food molecules. Also located in the submucosa is an extensive network of neurons known as the submucosal neural plexus. The submucosa may also contain glands and lymphatic tissue. The muscular layer of the mouth, pharynx, and superior and middle parts of the esophagus contains skeletal muscle that produces voluntary swallowing. Skeletal muscle also forms the external anal sphincter, which permits voluntary control of defecation. Throughout the rest of the digestive canal, the muscular layer consists of smooth muscle that is generally found in 2 sheets, an inner sheet of circular fibers and an outer sheet of longitudinal fibers. Involuntary contractions of the smooth muscle help break down food, mix it with digestive secretions, and propel it along the digestive canal. Between the layers of the muscular layer and the second plexus of neurons, we have something called the myenteric neural plexus. The serosa, those portions of the digestive canal that are suspended in the abdominal cavity, have a superficial layer called the serosa. As its name implies, the serosa is a serous membrane composed of areolar connective tissue and simple squamous epithelium. The serosa is also called the visceral peritoneum because it forms a portion of the peritoneum, which we will examine in detail shortly. The esophagus lacks a serosa. Instead, it has only a single layer of areolar connective tissue that's called an adventitia, and this adventitia forms a superficial layer of this organ. So let's move on to the neural innervation of the gut. The enteric nervous system, which is sometimes referred to as the brain of the gut, contains a submucosal plexus. This is found within the submucosa, and it also has a myenteric plexus, and this is located between the longitudinal and circular smooth muscle layers of the muscular layer. The autonomic nervous system also contributes to the digestive system, and although the enteric nervous system can function independently, it is subject to regulation by the neurons of the autonomic nervous system. So the parasympathetic preganglionic neurons of the vagus or pelvis-splanchnic nerves synapse with the parasympathetic postganglionic neurons located in the myenteric and submucosal neural plexuses. Some of the parasympathetic postganglionic neurons in turn synapse with neurons of the ENS, that means the enteric nervous system. Others directly innervate smooth muscle and glands within the wall of the digestive canal. In general, stimulation of the parasympathetic nerves that innervate the digestive canal causes an increase in digestive canal secretion and motility by increasing the activity of the ENS neurons. On the other hand, the sympathetic nerves that supply the digestive canal arise from the thoracic and upper lumbar regions of the spinal cord. Like the parasympathetic nerves, these sympathetic nerves form neural connections with the ENS. sympathetic postganglionic neurons synapse with the neurons located in the myenteric neural plexus and the submucosal neural plexus. In general, sympathetic nerves that supply the digestive canal cause a decrease in the digestive canal secretions and the motility of it by inhibiting the neurons of the ENS. Emotions such as anger, fear, and anxiety may slow the digestion because they stimulate the sympathetic nerves that supply the digestive canal. The GI reflex pathways. The GI reflex pathways regulate GI secretion and motility in response to stimuli within the GI tract. By motility, we mean that alternating contractions and relaxations of the smooth muscles in the wall of the digestive canal mix the food and secretions and move them toward the anus. This capability of the digestive canal to mix and move material along its length is called motility. Many neurons of the ENS are components of the digestive canal reflex pathways that regulate the digestive canal secretions and motility in response to the stimuli that's present in the lumen of the digestive canal. The initial components of a typical digestive canal reflex pathway are sensory receptors that are associated with the sensory neurons of the ENS. The axons of these sensory neurons can synapse with other neurons located in the ENS, the CNS, or the ANS, informing these regions about the nature of the contents and the degree of distension or stretching of the digestive canal. The neurons of the ENS, CNS, or ANS subsequently activate or inhibit digestive canal glands and smooth muscle, and this alters the digestive canal secretions and motility. And this is figure 24.5 on page 947. And this shows the peritoneum. The peritoneum is the largest serous membrane in the body. The peritoneum consists of a layer of simple squamous epithelium with an underlying supporting layer of areolar connective tissue. The peritoneum is divided into the parietal peritoneum, which lines the wall of the abdominal cavity, and the visceral peritoneum, which covers some of the organs in the abdominal cavity and is their serosa. So this figure shows the relationship of the peritoneal folds to one another and to the organs of the digestive canal. And in this cartoon, the size of the peritoneal cavity is exaggerated for emphasis. And this slide is a continuation of the previous slide. So this shows some of the gross anatomy of the peritoneum. And this slide is also a continuation of figure 24.5. And this is on page 948. And here we get the view from a cadaver. So the mouth is formed by the lips, cheeks, hard and soft palates of the oral cavity, teeth, salivary glands, and the tongue. And in this slide, we're looking at figure 24.6 on page 949. And this just shows the structures of the mouth. So moving on to the salivary glands. The salivary glands lie outside the mouth and empty their contents into ducts which deliver saliva into the oral cavity. And there's 3 pairs of salivary glands. You've got the parotid, the submandibular, and the sublingual. The parotid glands are located inferior and anterior to the ears between the skin and the masseter muscle. The submandibular glands are found in the floor of the oral cavity proper, and they are medial and partly inferior to the body of the mandible. The sublingual glands are beneath the tongue and superior to the submandibular glands. So this cartoon is figure 24.7 on page 950. As I said previously, there are three major salivary glands: the parotid, the sublingual, and the submandibular. The submandibular glands are shown in the light micrograph there in the bottom right, and they consist mostly of serous acini and a few mucous acini. The parotid glands consist of serous acini only, and the sublingual glands consist mostly of mucous acini and a few serous acini. So saliva lubricates and dissolves food and begins the chemical breakdown of carbohydrates and lipids. Moving on to the tongue. The tongue, together with associated muscle, forms the floor of the oral cavity. It's composed of skeletal muscle covered with a mucous membrane. It participates in chewing, swallowing, and speech. The upper and lateral surfaces of the tongue are covered with palpellae, some of which contain taste buds. So moving on to the teeth. This is figure 24.8 on page 952. The teeth are anchored in dental alveoli of the alveolar processes of the mandible and maxilla. And this cartoon shows a typical tooth and its surrounding structures. So please study this on your own time. And here we're viewing figure 24.9 on page 954. And this shows the dentitions and the time of eruption. So humans have two dentitions, or sets of teeth, the deciduous and the permanent. The first of these, the deciduous teeth, also called primary teeth, begin to erupt at about 6 months of age, and approximately 2 teeth appear each month thereafter until approximately 20 are present. Eventually, all of the deciduous teeth will be lost, and this generally occurs between the ages of 6 and 12 years, and then they will be replaced by the permanent teeth. The permanent dentition consists of 32 teeth that erupt between the ages of 6 and adulthood. The pattern resembles the deciduous dentition, with the following exceptions. The deciduous molars are replaced by the first and second premolars, which have 2 cusps and 1 root and are used for crushing and grinding. The permanent molars, which erupt into the mouth posterior to the premolars, do not replace any deciduous teeth and erupt as the jaw grows to accommodate them. So moving on to the mechanical and chemical digestion in the mouth. Mechanical digestion in the mouth results from chewing, or what we call mastication, in which food is manipulated by the tongue, ground by the teeth, and mixed with saliva. As a result, the food is reduced to a soft, flexible, easily swallowed mass called a bolus. Food molecules begin to dissolve in the water in the saliva, and this is an important activity because enzymes can react with food molecules in a liquid medium only. And in chemical digestion, the salivary amylase converts polysaccharides to disaccharides. So 2 enzymes, salivary amylase and linguolipase, contribute to the chemical digestion in the mouth. Salivary amylase, which is secreted by the salivary glands, initiates the breakdown of starch. Dietary carbohydrates are either monosaccharide or disaccharide sugars or complex polysaccharides such as starches. Most of the carbohydrates we eat are starches, but only monosaccharides can be absorbed into the bloodstream. Thus, ingested disaccharides and starches must be broken down into monosaccharides. The function of salivary amylase is to begin starch digestion by breaking down starch into smaller molecules such as the disaccharide maltose and the trisaccharide maltotriose, and short-chain glucose polymers, which we call alpha-dextrins. Even though the food is usually swallowed too quickly for the starches to be broken down in the mouth, salivary amylase in the swallowed food continues to act on the starches for about another hour, at which time acids inactivate it. Saliva also contains a lingual lipase, which is secreted by the lingual glands in the tongue. This enzyme becomes activated in the acidic environment of the stomach and thus starts to work after the food has already been swallowed. It breaks down dietary triglycerides, fats and oils, into fatty acids and diglycerides. A diglyceride consists of a glycerol molecule that is attached to two fatty acids. And this table 24.1 on page 955 summarizes the digestive activities in the mouth. So please study this on your own time. So this next cartoon takes us back to chapter 23, figure 23.3, and this slide is outlining the pharynx for us this time. So instead of talking about this cartoon in the respect of the respiratory system, now we're going to look at it in respect of the digestive system. The pharynx is a funnel-shaped tube that extends from the internal nares to the esophagus posteriorly and the larynx anteriorly. So this is figure 24.10 on page 956. The esophagus is a collapsible muscular tube that lies posterior to the trachea and connects the pharynx to the stomach. Also, the esophagus secretes mucus and transports food to the stomach. And if we look on the right of this figure, we can see a light micrograph of the layers of the esophagus. So this is figure 24.11 on page 957. And this talks about deglutition, or swallowing. During the pharyngeal stage, the tongue rises against the palate, the nasopharynx closes it off, and then the larynx rises. The epiglottis seals off the larynx, and the bolus is passed into the esophagus. During the esophageal stage, the food moves through the esophagus into the stomach via peristalsis. So deglutition is the mechanism that moves food from the mouth into the stomach. So this next slide is also part of figure 24.11. So we can see at step 1, in the section of the esophagus just superior to the bolus, the circular muscle fibers contract, constricting the esophageal wall and squeezing the bolus towards the stomach. At step 2, longitudinal muscle fibers inferior to the bolus also contract, which shortens this inferior section and pushes its walls outward so it can receive the bolus. The contractions are repeated in waves that push food toward the stomach. It should be noted that steps 1 and 2 will repeat until the bolus reaches the lower esophageal sphincter muscles. And then at 3, the lower esophageal sphincter relaxes and the bolus moves into the stomach. So table 24.2 appears on page 958. And this table summarizes the digestive activities in the pharynx and the esophagus. So please spend a little bit of your own time with this table. And this brings us to the stomach. And this is figure 24.12 on page 959. So we can see the external and internal anatomy of the stomach in this figure. The 4 regions of the stomach are the cardia fundus, the body, and the pyloric part. The stomach is a J-shaped enlargement of the GI tract. It functions to mix saliva, food, and gastric juice to form a soupy liquid called chyme. It also serves as a reservoir for food before the release into the small intestine. It also secretes gastric juice, which contains hydrochloric acid, pepsin, intrinsic factor, and gastric lipase. And fourthly, it secretes gastrin into the blood. And this is the same figure, but now we're looking at a cadaveric view of the stomach. And this figure appears on page 960, figure 24.13, and it goes through the histology of the stomach. Gastric juice is the combined secretions of mucus cells, parietal cells, and chief cells. So this next slide is also part of figure 24.13, but now we're on page 961. The stomach wall is composed of the same basic layers as the rest of the digestive canal, with certain modifications. The surface of the mucosa is a layer of simple columnar epithelium called surface mucus cells. The mucosa contains a lamina propria and a muscularis. Epithelial cells extend down into the lamina propria, where they form columns of secretory cells called gastric glands. Several gastric glands open into the bottom of narrow channels called gastric pits. Secretions from several gastric glands flow into each gastric pit and then into the lumen of the stomach. Gastric glands contain 3 types of exocrine gland cells that secrete their products into the stomach lumen: mucus neck cells, chief cells, and parietal cells. Both surface mucus cells and mucus neck cells secrete mucus. Parietal cells produce intrinsic factor and hydrochloric acid. The chief cells secrete pepsinogen and gastric lipase. The secretions of the mucus, parietal, and chief cells form what we call gastric juice. In addition, gastric glands include a type of enteroendocrine cell called the G cell, which is located mainly in the pyloric antrum and secretes the hormone gastrin into the bloodstream. As we will see shortly, this hormone stimulates several aspects of gastric activity. So this slide is a combination of figures 24.14 and 24.15, and these cartoons are found on page 962 and 963 of your textbook. But on the left-hand side, we see the secretion of hydrochloric acid by the parietal cells into the stomach. So proton pumps powered by ATP secrete hydrogen ions, chloride ions, and this diffuses into the stomach lumen through the chloride ion channels. On the right-hand side of the diagram, figure 24.15, we see hydrochloric acid secretion by parietal cells can be stimulated by several sources. Acetylcholine is one, gastrin is another, and histamine is yet another. And table 24.3 is also on page 963. So this is the summary of the digestive activities in the stomach. So please study this table on your own time. Moving on to the pancreas. The pancreas is a gland that lies posterior to the stomach. It produces enzymes that digest carbohydrates, proteins, fats, and nucleic acids. It also produces sodium bicarbonate, which buffers the stomach acid. And it empties its contents into the duodenum. So at the bottom of this slide, we see a pancreas from a cadaver, and this is figure 24.16 on page 965. So the pancreatic enzymes digest starches, or polysaccharides, proteins, triglycerides, and nucleic acids. This next slide is also figure 24.16, but now we're back to page 964. So this shows the relationship of the pancreas to the liver, gallbladder, and duodenum. And this slide shows a cadaveric view of the pancreas, liver, and gallbladder. And this is still figure 24.16, but now we're back to page 965. So moving on to the liver and the gallbladder. The liver is the heaviest gland of the body. Of all the organs of the body, it is second only to the skin in size. The liver is inferior to the diaphragm and occupies most of the right hypochondriac and part of the epigastric regions of the abdominopelvic cavity. The gallbladder, on the other hand, is a pear-shaped sac that is located in a depression of the posterior surface of the liver. It typically hangs from the inferior margin of the liver. So the liver makes bile, and that's important to the emulsification of fats, and the gallbladder stores bile until it's needed. And if we look at the right-hand side of this slide, we can see a flow diagram of how the liver and the gallbladder work in conjunction, and ultimately with the duodenum as well. So this is figure 24.17, and this is on page 967. And this talks about the histology of the liver. The liver is composed of hepatocytes, bile caniculi, and hepatic sinusoids. So the hepatocytes are the major functional cells of the liver and perform a wide array of metabolic, secretory, and endocrine functions. These are specialized epithelial cells with 5 to 12 sides that make up about 80% of the volume of the liver. The bile caniculae are small ducts between the hepatocytes that collect bile produced by the hepatocytes. From the bile caniculae, the bile passes into bile ductules and then into bile ducts. The bile ducts merge and eventually form the larger right and left hepatic ducts, which unite and exit the liver as the common hepatic duct. The common hepatic duct joins the cystic duct from the gallbladder to form the bile duct. From here, bile enters the duodenum of the small intestine to participate in digestion. Hepatic sinusoids are highly permeable blood capillaries between rows of hepatocytes that receive oxygenated blood from the branches of the hepatic artery and nutrient-rich deoxygenated blood from the branches of the hepatic portal vein. Recall that the hepatic portal vein brings venous blood from the digestive canal organs and spleen into the liver. Hepatic sinusoids converge and deliver blood into the central vein. From central veins, the blood flows into the hepatic veins, which drains into the inferior vena cava. In contrast to blood, which flows towards a central vein, bile flows in the opposite direction. Also present in hepatic sinusoids are fixed phagocytes. These cells destroy worn-out white and red blood cells, bacteria, and other foreign matter in the venous blood draining from the digestive canal. Together, a bile duct, branch of the hepatic artery, and a branch of the hepatic vein are referred to as the portal triad. And this next slide just shows some micrographs of the liver. Moving on to the next slide, this is figure 24.17 continued, and this is on page 968. The hepatocytes, bile duct system, and hepatic sinusoids can be organized into an anatomical and functional unit called the hepatic acinus. The hepatic acinus is an approximately oval mass that includes portions of two neighboring hepatic lobules. The short axis of the hepatic acinus is defined by branches of the portal triad, branches of the hepatic artery, vein, and bile ducts that run along the border of the hepatic lobules. The long axis of the acinus is defined by two imaginary curved lines, which connect to two central veins closest to the short axis. Hepatocytes in the hepatic acinus are arranged in three zones around the short axis, with no sharp boundaries between them. On this next slide, we see figure 24.18 on page 969. And this shows the blood supply of the liver. So the liver receives oxygenated blood via the hepatic artery and nutrient-rich deoxygenated blood via the hepatic portal vein. Functions of the liver and gallbladder. So in carbohydrate metabolism, the liver is especially important in maintaining a normal blood glucose level. When blood glucose is low, the liver can break down glycogen to glucose and release the glucose into the bloodstream. The liver can also convert certain amino acids and lactic acid into glucose, and it can convert sugars such as fructose and galactose into glucose. When blood glucose is high, as occurs just after eating a meal, the liver converts glucose to glycogen and triglycerides for storage. Concerning lipid metabolism, hepatocytes store some triglycerides, break down fatty acids to generate ATP, synthesize lipoproteins which transport fatty acids, triglycerides, and cholesterol to and from the body cells. It also synthesizes cholesterol, and it can use cholesterol to make bile salts. In protein metabolism, hepatocytes deaminate amino acids so that the amino acids can be used for ATP production or converted to carbohydrates or fats. What I mean by deamination is that the amino group is removed from the protein. The resulting toxic ammonia is converted then into a much less toxic urea, which is excreted in the urine. Hepatocytes also synthesize most blood plasma proteins such as alpha and beta globulins, albumin, protothrombin, and fibrinogen. The liver can also process drugs and hormones. So the liver can detoxify substances such as alcohol and excrete drugs such as penicillin, erythromycin, and sulfonamides into the bile. It can also chemically alter or excrete thyroid hormones and steroid hormones such as estrogens and aldosterone. The excretion of bilirubin. As previously noted, bilirubin, derived from the heme of aged red blood cells, is also absorbed by the liver from the blood and secreted into the bile. Most of the bilirubin in the bile is metabolized in the small intestine by bacteria and eliminated in the feces. The liver can also synthesize bile salts. Bile salts are used in the small intestine for the emulsification and absorption of lipids. The liver can also function as a storage unit. So in addition to glycogen, the liver is a prime storage site for certain vitamins and minerals, and these can be released from the liver when needed elsewhere in the body. Phagocytosis. The stellate reticuloendothelial cells of the liver phagocytized aged red blood cells, white blood cells, and some bacteria. And finally, the activation of vitamin D. The skin, liver, and kidneys participate in synthesizing the active form of vitamin D. The liver functions related to metabolism are discussed more fully in Chapter 25. So moving on to the small intestine, this is figure 24.19 on page 971. The majority of digestion and absorption occur in the small intestine. And in this cartoon, we see the regions of the small intestine are the duodenum, the jejunum, and the ileum. So moving on to the histology of the small intestine, this cadaveric image is on figure 24.20 on page 972. And this shows the circular folds that help to increase the surface area for digestion and absorption in the small intestine. And this is more specifically looking at the jejunum. Moving on to the next slide, same figure, same page. These circular folds, intestinal villi and microvilli, increase the surface area of the small intestine, and this helps with digestion and absorption. And this next slide is a variation on figure 24.20, the part that's on page 973. So we can see that the villi have blood capillaries that absorb amino acids, simple sugars, and other hydrophilics. Also, the villi have lymph capillaries called lacteals that absorb fatty stuff like hydrocarbons from the chyme. Intestinal juice and brush border enzymes. Intestinal juice provides a vehicle for absorption of substances from chyme as they come in contact with the villi. Brush border enzymes, found on the surfaces of the microvilli of absorptive cells, break down food products. So here we're looking at figure 24.21 on page 975 of your textbook. And this slide shows a micrograph of the wall of the duodenum. So the microvilli in the small intestine contain several microvillous border enzymes that help to digest nutrients. So moving on to the mechanical digestion in the small intestine. The two types of movement of the small intestine, segmentations and a type of paralysis called migrating motility complexes, are governed mainly by the myenteric neural plexus. Segmentations are localized, mixing contractions that occur in portions of the intestine distended by the large volume of chyme. Segmentations mix chyme with digestive juices and bring the particles of food into contact with the mucosa for absorption. They do not push the intestinal contents along the digestive canal. A segmentation starts with the contraction of circular muscle fibers in a portion of the small intestine, an action that constricts the intestines into segments. Next, muscle fibers that encircle the middle of each segment also contract, dividing each segment again. Finally, the muscle fibers that first contracted and relaxed, and each small segment unites with the adjoining small segment so that the large segments are formed again. As this sequence of events repeats, the chyme sloshes back and forth. Segmentations occur most rapidly in the duodenum, about 12 times per minute, and progressively slow to about 8 times per minute in the ileum. This movement is similar to alternating squeezing and the middle and ends of a capped tube of toothpaste. So after most of a meal has been absorbed, which lessens distension of the wall of the small intestine, segmentation stops and peristalsis begins. The type of peristalsis that occurs in the small intestine, termed a migrating motility complex, begins in the lower part of the stomach and pushes chyme forward along a short stretch of small intestine before dying out. The migrating motility complex slowly migrates down the small intestine, reaching the end of the ileum in about 90 to 120 minutes. Then another migrating motility complex begins in the stomach. Altogether, chyme remains in the small intestine for about 3 to 5 hours. So moving on to the chemical digestion in the small intestine. In the mouth, salivary amylase converts starch to maltose, maltotriose, and alpha-dextrins. In the stomach, pepsin converts proteins into peptides, and lingual and gastric lipases convert some triglycerides into fatty acids, diglycerides, and monoglycerides. Thus, chyme entering the small intestine contains partially digested carbohydrates, proteins, and lipids. The completion of digestion of carbohydrates, proteins, and lipids is a collective effort of pancreatic juice, bile, and intestinal juice in the small intestine. So that brings us to table 24.4 on page 980. And this table is a summary of the digestive activities in the pancreas, liver, gallbladder, and small intestine. So as usual, I will have you review this table on your own time. And in this next slide, we're looking at table 24.5, which appears on page 982. And this is a summary of digestive enzymes, so please review this on your own time. And this next slide is also a continuation of table 24.5. And this slide is also a continuation of 24.5. So like I said, please review these tables on your own time. They have a lot of useful information in them. So moving on to figure 24.22 on page 978. This concerns the absorption of digested nutrients in the small intestine. For simplicity, all digested foods are shown in the lumen of the small intestine, even though some nutrients are digested by the microvillus border enzymes. Long-chain fatty acids and monoglycerides are absorbed into the lymphatic capillaries. Other products of digestion enter the blood capillaries. All of the chemical and mechanical phases of digestion from the mouth through the small intestine are directed toward changing food into forms that can pass through the absorptive epithelial cells lining the mucosa and into the underlying blood and lymphatic vessels. These forms are monosaccharides from carbohydrates, single amino acids, dipeptides, and tripeptides from proteins, and fatty acids, glycerol, and monoglycerides from triglycerides. Passage of these digested nutrients from the digestive canal into the blood plasma or lymph is called absorption. Absorption of materials occurs via diffusion, facilitated diffusion, osmosis, and active transport. About 90% of all absorption of nutrients occurs in the small intestine. The other 10% occurs in the stomach and large intestine. Any undigested or unabsorbed material left in the small intestine passes on to the large intestine. So we're back to figure 24.22 now on page 978. And this shows the movement of absorbed nutrients into the blood plasma and lymph. So figure 24.23 on page 980. This shows daily volumes of fluid ingested, secreted, absorbed, and excreted from the digestive canal. All water absorption in the digestive canal occurs via osmosis. So moving on to the anatomy of the large intestine. The regions of the large intestine are the cecum, colon, and rectum. And this next slide discusses the functions of the large intestine. Haustral churning, peristalsis, and mass peristalsis drive the contents of the colon into the rectum. Bacteria in the large intestine convert proteins to amino acids, break down amino acids, and produce some B vitamins and some K vitamins. It also helps with the absorption of some water, ions, and vitamins. It forms the feces, and it acts in defecation or emptying of the rectum. So moving on to the histology of the colon. This is figure 24.25 on page 985. Intestinal glands formed by simple columnar epithelial cells and goblet cells extend the full thickness of the mucosa. The wall of the large intestine contains the typical 4 layers found in the rest of the digestive canal. That includes the mucosa, the submucosa, the muscular layer, and the serosa. The epithelium contains mostly absorptive and goblet cells. The absorptive cells function primarily in water absorption, and the goblet cells secrete mucus that lubricates the passage of the colonic contents. Both absorptive and goblet cells are located in long, straight, tubular intestinal glands that extend the full thickness of the mucosa. So this cartoon is part of the same figure, 24.25, and this just shows in greater detail the intestinal gland and its respective opening. It also shows a close-up view of goblet cells and absorptive cells. And on this next slide, we're still on figure 24.25, but we need to turn the page to page 986. And this slide shows us micrographs of the wall and the mucosa of the colon. So there on the right, you can see the goblet cell pointed out and the absorptive cell pointed out. And then on the left, the micrograph is divided into the mucosa, the submucosa, the muscularis, and the serosa, so all of that is clear for you to see. In haustral churning, distension reaches a certain point and the walls of the haustra contract to squeeze the contents onward. The haustra of the colon, singular haustrum, are small pouches caused by sacculation which give the colon its segmented appearance. In haustral churning, the haustra remain relaxed and become distended while they fill up. When the distension reaches a certain point, the walls contract and squeeze the contents into the next haustrum. Moving on to peristalsis. Peristalsis also occurs, although at a slower rate. And mass peristalsis, which is a strong peristaltic wave that begins in the transverse colon and quickly drives the contents of the colon into the rectum. Chemical digestion in the large intestine. The last stages of digestion occur through bacterial action. So substances are further broken down by bacteria, and some vitamins are synthesized by bacterial action. The final stage of digestion occurs in the colon through the activity of bacteria that inhabit the lumen. Mucus is secreted by the glands of the large intestine, but no enzymes are secreted. Chyme is prepared for elimination by the action of bacteria, which ferment any remaining carbohydrates and release hydrogen, carbon dioxide, and methane gases. These gases contribute to flatus in the colon, and it's termed flatulence when this is excessive. Bacteria also convert any remaining proteins to amino acids and break down the amino acids into simpler substances such as indole, scatol, hydrogen sulfide, and fatty acids. Some of the indole and scatol is eliminated in the feces and contributes to their odor. The rest is absorbed and transported to the liver, where the compounds are converted to less toxic compounds and excreted in the urine. Bacteria also decompose bilirubin into simpler pigments, including stercobilin, which gives feces their brown color. Bacterial products that are absorbed in the colon, absorption and feces formation. So in absorption, the colon absorbs water, electrolytes, and some vitamins. Feces consists of water, inorganic salts, sawed-off epithelial cells, bacteria, products of bacterial decomposition, and undigested portions of food. By the time chyme has remained in the large intestine 3 to 10 hours, it has become solid or semi-solid because of water absorption and is now called feces. Chemically, feces consists of water, inorganic salts, sloughed off epithelial cells from mucosa of the digestive canal, bacteria, products of bacterial decomposition, unabsorbed digested materials, and indigestible parts of food. Although 90% of all water absorption occurs in the small intestine, the large intestine absorbs enough to make it an important organ in maintaining the body's water balance. Of the 0.5 to 1 liter of water that enters the large intestine, all but about 100 to 200 milliliters is normally absorbed via osmosis. The large intestine also absorbs ions, including sodium and chloride, and some vitamins. The defecation reflex. Mass peristaltic movements push fecal material from the sigmoid colon into the rectum. The resulting distension of the rectal wall stimulates such receptors, which initiates a defecation reflex that results in defecation. Defecation is just the removal of feces from the rectum through the anus. The defecation reflex occurs as follows. In response to distension of the rectal wall, the receptors send sensory nerve impulses to the sacral spinal cord. Motor impulses from the cord travel along the parasympathetic nerves back to the descending colon, sigmoid colon, rectum, and anus. The resulting contraction of the longitudinal rectal muscles shortens the rectum, thereby increasing the pressure built within it. The pressure, along with voluntary contractions of the diaphragm and the abdominal muscles, plus the parasympathetic stimulation, opens the internal anal sphincter. If the external anal sphincter is voluntarily relaxed, defecation occurs and the feces are expelled. So table 24.6 appears on page 988, and it summarizes the digestive activities of the large intestine. So please review this on your own time. And in this next slide, we see table 24.7, which is also on page 988. And this summarizes the organs of the digestive system and their functions. So moving on to the phases of digestion. During the celiac phase of digestion, the smell, sight, thought, or initial taste of food activates neural centers in the cerebral cortex, hypothalamus, and brainstem. The brainstem then activates the facial, glossopharyngeal, and vagus nerves. The facial and glossopharyngeal nerves stimulate the salivary glands to secrete saliva, while the vagus nerve stimulates the gastric glands to secrete gastric juice. The purpose of the cephalic phase of digestion is to prepare the mouth and stomach for food that is about to be eaten. Once food reaches the stomach, the gastric phase of digestion begins. Neural and hormonal mechanisms regulate the gastric phase of digestion and promote gastric secretion and gastric motility. The intestinal phase of digestion begins once food enters the small intestine. In contrast to reflexes initiated during the cephalic and gastric phases, which stimulate the stomach secretory activity and motility, those occurring in the intestinal phase have inhibitory effects that slow the exit of chyme from the stomach. This prevents the duodenum from being overloaded with more chyme than it can handle. In addition, responses occurring during the intestinal phase promote the continued digestion of foods that have reached the small intestine. These activities of the intestinal phase of digestion are regulated by neural and hormonal mechanisms. So this figure is 24.26 on page 989. This figure shows the gastric phase of digestion, and it shows the regulation of gastric juice, pH, and gastric motility. So food entering the stomach stimulates the secretion of gastric juice and causes vigorous waves of peristalsis. So this next slide is table 24.8 on page 990. And this summarizes the major hormones that control digestion. So please review this table on your own time. Aging and the digestive system. This is on page 991. Aging results in the decreased secretory mechanisms and motility. It also results in a loss of strength and tone of the digestive muscular tissue. It changes in neurosecretory feedback. And it has a diminished response to pain and internal sensations. So here we have our focus on homeostasis, and this appears on page 994. So this shows how the digestive system contributes to homeostasis for all body systems. The digestive system breaks down dietary nutrients into forms that can be absorbed and used by body cells for producing ATP and building body tissues. It also absorbs water, minerals, and vitamins needed for growth and function of body tissues. And it also eliminates waste from the body tissues in the form of feces. Please review this on your own time. Digestive system disorder. Tooth decay or dental caries involves a gradual demineralization of the enamel of the dentin. If untreated, microorganisms may invade the pulp, causing inflammation and infection with subsequent death of the dental pulp and abscess of the alveolar bone surrounding the root's apex, requiring root canal therapy. Periodontal diseases. This is a collective term for a variety of conditions characterized by inflammation and degeneration of the gingivae, alveolar bone, periodontium, and cement. In one such condition called pyorrhea, initial symptoms include enlargement and inflammation of the soft tissue and bleeding of the gums. Without treatment, the soft tissue may deteriorate and the alveolar bone may be reabsorbed. This causes loosening of the teeth and recession of the gums. Periodontal diseases are often caused by poor oral hygiene, by local irritants such as bacteria, impacted food, cigarette smoke, or by a poor bite. And what I mean by bite is how your teeth line up when you close your mouth. Ulcers. An ulcer is a crater-like lesion in a membrane. Ulcers that develop in areas of the digestive canal are exposed to acidic gastric juice, and these are called peptic ulcers. Diverticular disease. This refers to sac-like outpouchings of the wall of the colon. They occur in places where the muscular layer has weakened and may become inflamed. Development of diverticula is known as diverticulosis. Many people who develop diverticulosis have no symptoms and experience no complications, but of the known people who have diverticulosis, 10 to 25% of them eventually develop an inflammation known as diverticulitis. Colorectal cancer is among the deadliest of malignancies, ranking second to lung cancer in males and third after lung cancer and breast cancer in females. Genetics play a very important role, and inherited predisposition contributes to more than half of all cases of colorectal cancer. The intake of alcohol and diets high in animal fat and protein are associated with an increased risk of colorectal cancer. Hepatitis is an inflammation of the liver that can be caused by viruses, drugs, and chemicals, including alcohol. Clinically severe types of viral hepatitis are recognized. Hepatitis A is spread via fecal contamination of objects such as food, clothing, toys, and eating utensils. Hepatitis B is caused by the hepatitis B virus and is spread primarily by sexual contact in contaminated syringes and transfusion equipment. Hepatitis C is caused by the hepatitis C virus and is clinically similar to hepatitis B. Hepatitis C can cause cirrhosis and possibly liver cancer. Hepatitis D is transmitted like hepatitis B, and in fact, a person must have been co-infected with hepatitis B before contracting hepatitis D. Hepatitis E is caused by the hepatitis E virus and is spread like hepatitis A. Although it does not cause chronic liver disease, hepatitis E has a very high mortality rate among pregnant women. And that concludes tonight's lecture.